So the title of the talk is Cybernetics, a Conversation with the Cosmos, which sounds like a mouthful. I didn't want to neglect to mention that. So my training is as a cell biologist. I spent 50 years doing experimental cell biology in the context of developmental physiology. And about 20 years ago, I devised a schematic for a review paper I was writing on the development of the lung alveolus, the gas exchange site, the little saccules in our lungs, and came to the realization that either what I was showing a schematic for was was neutral theory, Fukuyama's idea that things will happen over given enough time, or there was some underlying set of principles that was creating these 40 different cell types from basically the three germ layers, the endoderm, ectoderm, and mesoderm. And when I did the statistics, turns out that it would have taken longer than the age of the universe, longer than the age of the universe for the lung to have evolved purely by chance. So the alternative was that there was some underlying set of principles involved. I also wanted to mention that in my musings about cellular evolution, I came to the realization that the first cell was formed based upon Schrodinger's idea, in my opinion, of negative entropy. And so you have the origins of life being a paradox, if you will, the paradox of internal negative entropy and external positive entropy. In the interim, from the origins of life several billion years ago to the present day, we've made up all kinds of narratives. We tell ourselves stories about how we evolved, whether it's religious or mythical or everything in between. And the premise of my talk is that I think we have to move on to a narrative that actually is based upon science, and that it would be, in Popper's terms, testable and refutable. So basically everything I'm going to talk about, short of quantum mechanics and physiology, is all based upon experimental evidence. And in fact, the quantum mechanical idea actually is predicated on an experiment I did 20 years ago with microgravity affecting cellular physiology. So even that is under the comes under the auspices of testable and refutable. At any rate, if we could have the next slide. And please, if you have questions, interrupt me, because this is a lot of material and it's fairly esoteric and eclectic. So on the left is what I was talking about, this diagram that I devised. On the far right is the microvasculature of an alveolus, which is on the far left of the airspace. And then you have these two cell types that are interacting with one another. And they basically keep us alive. They maintain the patency, the opening state of the alveolus. Without this cell-cell signaling mechanism, for example, newborns that are born prematurely will die because they don't have the full complement of these cellular mechanisms. It's called hyaline membrane disease or respiratory distress syndrome, and that's basically how I came into onto the scene, if you will, to ask these scientific questions, because it had been discovered back in the late 60s that a simple molecule, cortisol, it's relatively simple, accelerates this whole process that I'm showing you here in this diagram physiologically, such that babies who were born prematurely no longer died. Back in the day, I mean, the Greeks used to take premature infants and just put them on a rock, and either they survived or they died. We're a little bit more sophisticated now, but and most children, virtually all of them now survive through recognition of the administration of cortisol to the mother, threatening preterm labor, and postnatally after birth by squirting lung surfactant into the airway. Surfactant is a surface tension reducing factor which is produced within the alveoli, and these premature infants are deficient. So on the right is the way we normally think of lungs, and then that little inset down in the lower right corner is blown up in the left-hand diagram. Okay, so the alveolus. So as I said, there are 40 different cell types in the lung. And so the question is, how did this transpire? How did we form a lung? And we now know that the lung evolved from the swim bladder of fish. So the swim bladder is an organ also an organ of gas exchange, which should be kept in mind. But in the case of the fish, it maintains buoyancy. It allows the fish to determine whether it's going to sleep at the bottom of the body of water or somewhere in between the surface and the bottom for feeding purposes. So it increases efficiency of the physiologic system. So Lynn Margulis was a professor of mine at Boston University, and Lynn was the one who fostered the idea of symbiogenesis. That is to say that the way that cellular evolution and physiologic evolution has transpired is that so unicells dominated the Earth for the first 3.5 billion years. It's only within the last 500 million years that multicellular organisms have evolved. And Margulis's symbiogenesis theory is that when unicellular organisms initially were confronted with an existential threat, what they would do would be to internalize that agent or that factor. So the primary example is bacteria being internalized within our cells to form mitochondria, which generate energy. But there are many, many examples. Every physiologic trait that we have evolved from that kind of phenomenology. So for example, iron is a very potent oxidizing agent in the environment, but in the red cells of our body, in the heme protein, it's the core of the heme protein that allows for oxygen to be carried around through the circulation and exchanged with cells for metabolic function, for example. Okay, so as I say at the bottom there, so Margulis' symbiogenesis theory is that we evolved through internalized environmental factors, which merged biology and physics as a monistic perspective. This idea of monism is really neglected in the literature. It, for example, accounts for Descartes' mind-body dichotomy, which is an artifact. The reality is that, as my co-author, Bill Miller, is fond of saying, it's like turning your sock inside out. You have to realize that there was this critical juncture at which biology and physics merged together. And we have to think of biology from the perspective of physics. And in fact, I'll talk about not only physics per se, but quantum mechanics as well. So next slide. So on the upper left-hand corner there, I show a diagram. So the right side of that panel, there's a stippled line that runs down the middle. On the right-hand side is the same diagram I showed you in the first slide. On the left is this idea that development actually recapitulates the process of symbiogenesis. I'm showing that diagrammatically. And furthermore, development and phylogeny, speciation, are virtually the same process because both of them are dependent upon cellular communication for the formation of structure and function. And so I'm showing to the right, there are two diagrams. One is of an alveolus in the lung, the other is a glomerulus in the kidney. So in both cases, the same molecular signaling pathways determine either gas exchange in the case of the lung alveolus, or the exchange, if you will, of fluids and electrolytes in the kidney between the vasculature, the capillaries in either case, and the kidney tubule in the case of the kidney. So basically, these are what are called pliotropic mechanisms. They're the same mechanism used for two different purposes. So this is what Gould and Verba called exaptation. And it's the way that I was able to deconvolute the lung alveolus all the way back to this unicellular state, for example, going backwards in time, space-time, both structurally and functionally, based upon both developmental and phylogenetic, and in many cases, pathological conditions, because those recapitulate evolution, if you will. So I'm claiming that, so now I'll also describe this diagram on the lower left. I did a back of the napkin kind of drawing at one stage and thinking about this kind of thing. And I realized that if I regressed the genes that generate the lung alveolus against geologic epochs, if you will, shown on the x-axis, below the x-axis. So for example, going from fresh to salt water, from water to land, which was critically important in our evolutionary physiology, or as it's now known, it's referred to as the burner hypothesis, oxygen didn't go from 0 to 21% oxygen, which is the prevailing oxygen tension in the atmosphere. It actually went up and down fairly dramatically between 15 and 35%. And we all, I think, are aware of the effects of increased oxygen in the atmosphere. That's what generated gigantic insects and dinosaurs. But no one to date, other than myself, has addressed the downside of all that. That's the decrease in oxygen tension, which occurred, which is referred to physiologically as hypoxia. Hypoxia is the most potent stimulator of the hypothalamic pituitary adrenal axis, stress. And so I've introduced the idea that stress, periodic stress, has actually generated various structural forms during land adaptation. So as I say, each step in the progression is a Gould-Verba exaptation, or what's referred to as a pre-adaptation, which explains the concept of emergence. How is it that things that didn't exist can emerge from nothing, seemingly? Well, that's not the case. They emerge from the genetic composition of the organism, and how, as I was showing here with the alveolus and the glomerulus, how there was this reattribution of pre-existing genetic traits for novel purposes. That's really what emergence is. So next slide, please. And so on the upper left, I'm showing this idea of how ontogeny and phylogeny as a vector were actually generated by this selection pressure that refers all the way back to the Big Bang, and that in turn references the singularity. So in my opinion, the unicellular state is actually referencing the singularity because that's the prototype for the generation of the first cell. And as I say here, the micelle is a protected space. Micelles are generated by immersing lipids in water. And we know that lipids were present on asteroids that were made of frozen water that generated the primordial ocean. So there were lipids on those asteroids as well. And those lipids are generated by pulsars out in deep space. So again, the micelle is a protected space that allows entry of particles under the influence of quantum entanglement, is my hypothesis, which referenced the non-local tandem events in the cosmos. So this is quantum mechanics. This quantum mechanical relationship between life and non-life is the basis of evolution. So what I'm saying is, and the reason that I feel strong, and the other concept that's embedded here in this diagram is that in reality, those cellular communications that promote both development and phylogeny are actually high energy phosphates. So as the one cell produces a soluble growth factor that binds to a specific receptor on a neighboring cell, the neighboring cell will generate a second message, is what the jargon is in my field. And that second message, first discovered by Earl Sutherland back in the 60s, for which he won the Nobel Prize. He discovered that cyclic AMP was an important second messenger. That is a high energy phosphate. So in reality, the embryologic changes that we are familiar with, going from a fertilized egg, a zygote, to a morula, to a gastrula, et cetera, et cetera, et cetera, those are all really just phenotypic changes that allow for the organism to interface with the environment ultimately over the course of development, and prior to that, the phylogenetic changes that have occurred. So again, those are all high energy phosphates. And so in reality, we make a systematic error in thinking that evolution and that our being is actually material. It's actually energy waves that were generated by the Big Bang, producing, so if we think about nucleosynthesis, for example, how stars generate light, they actually generate light by producing matter. And that matter starts with hydrogen and goes all the way to uranium and everything in between based upon atomic mass. So the stars are actually producing the logic of the cosmos, because it's based, for us anyway, the periodic table, for example, is essentially an algorithm for that relationship between the atomic mass and its order within the cosmos. And through symbiogenesis and the internalization of these factors, specifically the elements of the table, we also comply with the same laws of nature that the cosmos does. So we're basically, the cosmos and our consciousness is operating off the same set of rules. That's what consciousness actually is. So physiology is an algorithm for that. And that is a way in which we can understand what consciousness actually constitutes. It's not something separate from us. It's not mind. It's not brain. Brain is just a concentration of neurons. In reality, the peripheral nervous system gave rise to the central nervous system. So anecdotally, there was a big problem in the study of the evolution of the central nervous system because people couldn't figure out how we got from invertebrates without a backbone to vertebrates because there was a gap there until Nick Holland published his paper back in the 80s on the skin brain hypothesis, which is that invertebrates have a central nervous system. It's just that it's not centralized in a head structure as a brain. It's peripheral. It's a peripheral nervous system. So there is this relationship between the peripheral nervous system and the central nervous system that we must recognize. And it's the peripheral nervous system, for example, which answers David Chalmers' question, the hard question of psychology. Why is it that when we whack our thumb with a hammer, we see red? I maintain that that's because it's an atavistic trait that's referencing a stage in our physiologic evolution when we were able to develop a closed vasculature and a nervous system, which when you were injured, you would bleed and you would feel pain. So that's really where the hard question is answered, in my opinion. So then the question, so I'm saying that all of this is equivalent to cybernetics as the homeostatic feedback from the cosmos. I chose to focus on cybernetics for this group because my work is so potentially distant from the kinds of things that you folks discuss on a regular basis. But I did come to the realization by watching your end of 2020 Festschrift that a lot of you are cyberneticians. And so I thought I'd couch it in cybernetic terms. And I might say, with all hubris aside, this is the power of this approach to understanding biology and physics, because it is universal. It can be, in my opinion, it can be expressed in terms of cybernetics. First order cybernetics as a feedback mechanism, second order von Forster recognition of that, the third order being these cell-cell communications, which came after von Forster and Wiener for that matter. They were discovered in my laboratory when I was a grad student and other labs as well. And then finally, you have fourth order cybernetics, which I'm claiming is quantum mechanical. Next slide. So I just wanted to spell out this idea of how our origins as lipids, shown at the bottom of this upward cascade. So lipids are what are called amphiphiles. They have a positive and negative charge. And because of that, when they're immersed in water, they will form perpendicularly to the surface of the water, and they will pack. And the packing actually reduces the surface tension of water, which is actually quite high. Van der Waals forces are quite powerful. So, if you put a transparent straw into a glass of water, that meniscus that's formed, that's surface tension. And it has to be reduced in order for life to actually form effectively. And so that's what I'm showing here is that as we go up to the next level, these cells will spontaneously form micelles. Those micelles or liposomes. So just for a reality check, when you get your COVID injection, the mRNA for the spike protein of COVID is actually injected into you in what are called liposomes. They can be produced in the laboratory by wetting and drying lipids recursively, and eventually those lipids will form spheres. That's what I'm showing here. And then calcium is being leached from the bedrock, if you will, because carbon dioxide produced by primitive plants dissolves in water, forms carbonic acid, and that leaches calcium from the bedrock, for example. So calcium is rising in the primordial ocean. And as these lipids deform under the warming effects of the sun, because they will entrap calcium more and more so, but because lipids express this characteristic of hysteresis, they have molecular memory. When things get cold at night, they will reform in their original configuration, but the calcium is being trapped over and over again. And eventually, there are a subset of these micelles which were able to form calcium channels to control the amount of calcium in and out of the cell. That is our origin. So this is where we have originated from. And I might point out that verticalization of the lipids down at the bottom, actually, in my opinion, is the atavistic reference to why we stand on two legs and are bipedal, for example. So these are the kinds of long arc kinds of relationships, atavistic traits that are always referencing the first principles of physiology, physically and behaviorally as well. So next slide, please. So the idea was that we know that we evolved from fish, from bony fish. Specifically, there are two classes of bony fish. There are Physostomus and Physoclystis fish. That's based upon how their swim bladder is filled and emptied. So in the case of Physoclystis fish, their way of doing that is somewhat like a kidney. So gases in the circulation are extracted into this bladder or expelled back into the vasculature. That's not where we evolved from. We evolved from Physostomus fish, which actually have a pneumatic duct that connects the esophagus of the fish to the bladder. It's totally molecularly and cellularly and functionally homologous with our lungs. So it's a specific organism. And we know that through Neil Shubin's work that there is the fossilized evidence for this phenomenon. It's called Tiktaalik. This organism is a tetrapod, four-legged animal that crawled out of the ocean and onto land, primarily because, first of all, land masses were exposed because of what's called the greenhouse effect. This is Romer's hypothesis. And furthermore, the elevation of the greenhouse effect, as we're experiencing now, raised the temperature of the atmosphere and caused a decrease in oxygen in the dissolved oxygen in the ocean, because the amount of dissolved gas in the ocean is determined by temperature. And so there was a decrease in oxygen and there were these land masses. So there were these bony fish that decided, if you will, to crawl out into land. But nobody has addressed how they accomplished that in terms of their internal organs. That literature is only contributed to by me. Okay. And furthermore, there were 3 gene duplications that occurred during the water to land transition. We know that for a fact. The gene that I studied for 30 years under NIH funding was parathyroid hormone related protein, which if you delete that gene in a mouse, it will die at birth because it has no alveoli. So clearly it was fundable and was important and remains so. So, but picture this. So you have this organism that's crawling out of water and testing, land, if you will. It's still got a swim bladder, and it's evolving this lung structure from the swim bladder on a periodic basis. And these cell-cell communications are what's causing an internal remodeling of this structure and function. And the way that works is, so you have cellular mechanisms that determine either gas exchange or kidney function or skin function, et cetera, et cetera, et cetera. And when they're under duress, under stress, these cells will lose their homeostatic set point. And the individual cells will generate what's called radical oxygen species, which are a response to that physiologic stress. And those radical oxygen species are known to cause gene mutations and duplications. That's where that gene duplication process came from. And so I'm showing here, starting with the lung, you have a condition of hypoxia, intermittent hypoxia from time to time, in which the lung is not able to maintain the metabolic function of the organism. And that fires the pituitary adrenal axis to produce adrenaline. So you have the pituitary at the top of that image that causes an increase in adrenocorticotropic hormone. That in turn stimulates the adrenal gland on the right to produce cortisol. And cortisol then goes down through so there are 2 components to the adrenal gland. There's the triangular shape is where the cortisol is produced. And then that rectangular shape is the medulla. That's where adrenaline is produced. And so when cortisol is produced in the triangular structure, it filters down through that rectangular structure and stimulates the rate limiting step in adrenaline production. And so adrenaline then goes into the circulation and actually releases that constraint on the lung, the inability to exchange enough oxygen, because the adrenaline causes a secretion of that lung surfactant shown in tandem with the oxygen goes down. Now surfactant levels go up in the alveoli, allowing for the alveoli to stretch out further and to increase gas exchange. So you have an alleviation of the constraint on the lung, and that stretching mechanism actually stimulates this PTHRP mechanism to form more alveoli over the long haul, but acutely it allows for increased oxygenation. In tandem with that, adrenaline also will stimulate what's referred to as lipolysis, the release of fatty acids from fat cells in the body. That produces body heat because fatty acids are the most efficient source of calories, if you will, for the generation of body heat. And so the body heat goes up, and eventually this ad hoc kind of Rube Goldberg kind of system then is replaced by, in the case of humans, of mammals, oxytocin produced by the posterior pituitary, and that is the genetic determinant of body temperature. All of that in turn gave rise to bipedalism because warm-blooded organisms are much more efficient at metabolism than cold-blooded organisms. So it takes like 5 different enzymes to accomplish any one metabolic step in a cold-blooded organism, because it has to be just, Goldilocks effect has to be just right for the temperature that the atmosphere is at. Whereas in warm-blooded organisms, there's only one enzyme necessary. And so that increases the efficiency in order to then, in our case, stand up on 2 legs, for our forelimbs for tool making and for language being a tool making process. That in turn created selection pressure for the central nervous system. And as Simon Conway Morris is fond of saying, first there were bacteria, now there is New York. Well, how did that happen? This is the origin of that. That is to say that the selection pressure for the central nervous system generated our large heads and imagination, and the combination of tool making and language and imagination generated New York. Next slide. So I'm showing that cascade. I sort of preempted myself, but if you start at the very bottom with this water to land transition and physiologic stress generating endothermy or warm-bloodedness, that in turn gave rise to bipedalism. And I'm referencing the perpendicularity of the lipids as being the antecedent of that. And the combination of bipedalism and increased body temperature and regulated body temperature were atavistic traits, freeing the forelimbs for tool making and language. And the reason I'm confident in saying that tool making and language come from common sources is because all great apes have what's called the area of Broca within the cerebrum. We, including us, the difference between us and all other great apes is that we stand on 2 legs and we have the freedom of our forelimbs to do these special effects. That's the critical difference that caused that selection pressure for tool making, language, civilization, et cetera. So again, that positive selection for the central nervous system generated our oversized heads, led to what's in clinical obstetric terms, led to cephalopelvic disproportion. So there's this inability of the fetus to get through the birth canal. That is why we are born with only about 25% of adult brain capacity or volume. And so it takes 20 or 30 years, if ever, for humans to mature. And during that process, you have all these epigenetic mechanisms that contribute to who we are as a species. But my point is that we are unique in that regard because we require this huge amount of time in relationship to our immediate family, to our local environment, and then based upon these cellular molecular mechanisms, basically. And so there is this arc from the unicell all the way to Gaia, Lovelock's idea of Mother Earth as a continuum. And so we basically are juvenile great apes. And other people have suggested this, but they've never provided a mechanistic explanation. I've provided that. And I'm suggesting, I will propose that it's important that we understand where we originate from, how it is that we continue to tell ourselves false narratives, but that we as a species are running out of time and resources, and we must revert to a narrative that is faithful to our scientific knowledge base. So without sounding too preachy, I think that the time has come for us to make this paradigm shift that I'm asking for. And thanks for your attention, and I certainly appreciate any questions and comments.
Questions for clarification before the slides being turned off? No questions.
No questions. Too many questions to ask. So don't know which one to ask.
A lot of questions.
First one.
The top one, the burning one.
Yeah, actually, I had a conversation with John already about a week ago. Actually, he also sent me a couple of articles to read, so I had some kind of a preparation. I think that's extremely helpful. I think I actually learned a lot from John's articles. It is very basic ideas of negative entropy and how that entropy is protected by a membrane, a cell intended homeostasis within the membrane, and how the complexity can be increased and life eventually evolved on that basis. So I thank you very much. Actually, I was talking to Jason the other day about probably allowing John to talk more, but Jason said, okay, we can't allow each person to talk too long, so the discussion will not be impossible. So I think.
well, not that serious. John, I posted a message to our group saying that who will be making some connections between the self-organization framework that this group are very familiar with, such as Ashby's frame, Heinz von Foerster's frame, Prigoghine's frame, and Herman Hawkins' frame. And you send me a message, answer that question. Could you please share that idea with us as well?
Sure. Yeah, I'm more familiar with Maturana and Varela's autopoiesis, but we're talking about the same phenomenon. So yes, I think what I'm talking about is a mechanistic explanation for how and why we are self-referentially, self-organizing, self-authoring organisms. So I think in 2017, there were 2 independent laboratories who showed that yttrium atoms will actually associate with one another spontaneously, for example. So this was a physical example of how autopoiesis exists within the physical world. And because I'm predicating what I'm saying here on the fact that biology is the internalization of those physical factors, that's why we in turn are self-referentially, self-organizing, self-authoring organisms. It's predicated on the same natural law. And my guess, and I know it's controversial as to whether to hang your hat too heavily on the Big Bang, but basically my gestalt is, the Big Bang generated all these asymmetries, and that homeostasis, which is central to both the physical and chemical balanced equations as well as biology, that homeostasis is really is Newton's third law. It's the equal and opposite reaction that is being strived for. The difference between biology and chemistry and physics is that we have been manned with having to maintain equipoise with an ever-changing environment. Physics and chemistry, they actually do the same thing, but the timelines are geologic, if you will. But they do go through the same process of creation and degradation and then recreation that we do. It's just that the timelines are different. So, Teilhard de Chardin was famous for saying that a rock has consciousness. It's just zero. Yeah, and I agree with that. So I don't know if that answers your question, but that's how I perceive the self-referential, self-organizational issue.
Lloyd, you're muted. You're still muted.
I hope I'm not unmuted now.
Yeah, you're okay now.
Hi. Yes, thanks for the presentation. If I correctly understood, but I'm not sure I did, it's rather reductionist, isn't it? But then at the biological level, there's some self-organizing potential in which I would say that the emerging order tends to take over the underlying orders, which have been the morphogenesis of the system. Perhaps I'm talking in a very different type of wording than you are used to, maybe not. If there is such a feedback arrow, which can take over control, the dependency between the emerging level and the level, the reductionist level, is not necessarily bottom up, but it can also be top down. And I think in human societies, for example, we have this very strongly. For example, monetary transactions are very regulating on our behavior. It's just an example. So you get a top-down error. Is that not a tension with your claim that you can explain everything bottom up?
I'm actually, thanks, Luke, for the question. I actually think of it as middle out. Sid Brenner, the Nobelist, was interested in the middle out approach also, but his answer, his experimental model was, he was trying to determine all of the contents of the cell. But the problem with that is then there has to be some set of operating principles. That's where I come in and I say, okay, no, the cell is not acting independently. Oh, is independent, sure, but it's in the case of unicells, it's reacting to the environment that it's in. In the case of multicellular organisms, it's responding to the cells around it, but it's through these cellular communication processes. So you're absolutely right. I mean, I have been a reductionist for most of my career because that's how I got granted. I was funded. You had to be reduction. But in reality, 50 years after the fact, I had the opportunity to think about the holistic way of thinking about this. And so, yeah, I agree with you. I think that it's both reduction, the reductionism led to enough pieces of the puzzle that then the Humpty Dumpty could be put back together again, essentially based upon these cellular communication mechanisms, which are the way in which to then diachronically. So, Bohm's, there's both a synchronic approach and a diachronic approach. I find that the synchronic approach is the common way of thinking about these things as how to fit things together in real time, space and time. However, in order to strip away all the material epiphenomena, you have to transcend space and time, which is the diachronic approach. That's what I'm trying to express here.
I would like to go one step further, if I may, Jason.
Yeah.
To say that, if there is an emerging level, then there is no necessity to stop at the biological level. There may be also psychological mechanisms, which are of a different kind, and also sociological, cultural mechanisms, which are very different. And you cannot explain that from the biological level because there are too many in-between levels.
Well, with all due respect, so I published a paper, I came across, the niche construction concept, and I thought to myself, this is simplistic and it's really not helping me. But then I thought, oh, well, and based upon symbiogenesis, well, maybe the unicell was the first niche construction. Now you have the internal interstice, the internal workings of the cell functioning off the ecologic system that it exists in. So there's this, it's Bohm's enfolding and unfolding. And that allows you to go from a cellular molecular perspective all the way from the unicell to Gaia, in my opinion. But in terms of the psychology and the sociology, so I'm accounting for the sociology that way. But in terms of the psychology, I published another paper based upon our work on epigenetic inheritance, the realization that, so we work on how maternal and paternal cigarette smoking causes childhood asthma. That's the specific problem we're dealing with. But what we've shown is that if you treat a mother rat during pregnancy with nicotine, the offspring will have asthma for at least 3 generations. We couldn't go beyond that because it was too expensive to do. But in humans, it's certainly it's a totally consistent with what we see in humans as well. The reason we did the studies to begin with 15 years ago, beginning of 15 years ago, was a group at USC had shown that it wasn't a function of whether your mother smoked or your father smoked, it's whether your grandmother smoked was the biggest signal. So that gave that insight to the transgenerational effect of nicotine. But there are many, many examples of that are coming out. But that's actually direct acquisition of information, physically and then chemically from the environment. And it can be behavioral also. But my point being that, so I wrote a paper on the phenotype not being a laundry list of traits, but in fact, the phenotype being an agent. It's acting as an agent to obtain information from the environment, looking for novelty in essence. So for example, we know that the endocrine system is under epigenetic control. For example, that's a huge observation because it addresses your comment about psychology, behavioral aspects. So yeah, I don't know if that answers your question, but that's how I do it.